Okay, today we are going to study about placenta formation, how the placenta is formed. After the formation of diploid zygote, which happens immediately after fertilization, when the sperm and haploid nuclei of sperm and egg they fuse together, then they form diploid zygote. Then this diploid zygote will keep on dividing by mitosis. What is mitosis? It is a type of cell division in which occur in normal body cells in which number of chromosomes remains same. Okay, so it will keep on dividing by mitosis and form embryo. Now this embryo will keep on moving, it will keep on dividing, it will keep on moving towards the uterus by the beating movement of the cilia, and then it implant itself in the uterine lining. The embryo will implant itself in the uterine lining. Now embryo will keep on dividing, the sum of the cells of the embryo, they will keep on dividing and it will form the finger-like projections. This finger-like projection is known as embryonic villi, and within this embryonic villi there lies embryonic capillaries. This embryonic villi along with the uterine lining is collectively known as placenta. So what is placenta? Placenta is this structure, embryonic villi plus the uterine lining in which it is embedded. It is collectively known as what? Placenta. Okay. So we can say that placenta is made partly from the embryonic tissue and partly from mother tissue. Of embryonic tissue there lies embryonic villi, and from mother tissue there lies uterine lining. In the uterine lining there is a blood filled space. Okay, in the uterine lining there is a blood filled space. Okay, so what is the function of this placenta? It allows the diffusion of oxygen and dissolved food substances. You have to give two examples, at least two examples of dissolved food substances. Dissolved food substances, for example, glucose and amino acids from the mother's blood spaces into the fetal blood capillaries. Okay, and then first point, and then it allows the removal of carbon dioxide, urea and uric acid, of course, from where to where? From from embryonic villi into the mother's blood spaces. Second point. And the third point, it secretes progesterone. Now what is the function of this progesterone? Progesterone, it maintains the thickness of uterine lining. It maintains the thickness of uterine lining throughout the gestation period or throughout the pregnancy. Okay, it maintains the thickness of the uterine lining, because we know that the placenta formation, it happens in the uterine lining, and if there is no maintenance of progesterone level, then what happened? We know that progesterone, it maintains the thickness of uterine lining, so uterine lining will break down and it will come out through vagina and the menstruation will begin. Okay, so in order to keep the pregnancy, the secretion of progesterone is vital. Okay, so what is the function of progesterone? It maintains the thickness of the uterine lining. Now as you can see over here, this whole structure is known as placenta. Now this structure, which is connecting the fetus with placenta, is known as umbilical cord. What is the function of umbilical cord? Umbilical cord connects the fetus with the placenta. Now, what is the function of this umbilical cord? It will allow the diffusion of oxygen, glucose and amino acids from the placenta to the fetus. So, the oxygen, glucose and amino acid which are diffused from the mother's blood spaces, at first it gain entry into the embryonic villi, into the blood capillaries which are present in the embryonic villi, and then the embryonic villi will unite and form what? Embryonic will form what? Embryonic vein, or which is called umbilical vein, and then the umbilical vein will transfer the glucose and amino acid to the fetus. This umbilical vein are present inside umbilical vein is present inside the umbilical cord. It contains one umbilical vein and two umbilical arteries. Now, what is the function of umbilical artery? The function of umbilical artery is to carry urea, uric acid and glucose, and carbon dioxide from the fetus to the placenta, or you can write from the fetus into the embryonic blood capillaries which are present in the embryonic villi, and then from the embryonic villi it will diffuse outside in the maternal blood spaces. Now, one more important thing is blood entering the placenta from the mother does not mix with the embryo's blood. Now, why it is important? It is important because the mother and embryo may have different blood group. So, as the mother and fetus may have different blood group, so it may cause agglutination. Fine. Number one. Number two, why it is important? Because high blood pressure of the mother may kill the fetus. Okay, so why blood entering the placenta from mother does not mix with the embryonic blood? Why it is important? Number one, because the mother and embryonic blood may have different blood group, so it may cause agglutination. We know that when there are two different blood group, then it may lead to agglutination or clumping of the blood. And number two, the high blood pressure of the mother may kill the fetus. And one more thing, it prevents the entry of harmful substances. One another function of the placenta, it prevents the entry of harmful substances into the fetus. But sometimes some viruses may gain entry across the placenta into the fetus. That is the AIDS virus, HIV virus. It can gain entry from the placenta into the fetus. Okay.